golden age of microbiology is a period of about 50 years that occurred at the late 1800s and into the 1900s. And this is right about the time when the term microorganism was actually coming into use. So there were a number of things that sparked this golden age of microbiology, or this great rush of scientists to study all things microorganism. So there were several questions that scientists were actually seeking answers to. And the seeking of answers to these questions is why scientists were actually studying microbes so closely. So first off, scientists wanted to know, is spontaneous generation of microbial life possible? So basically, can we evolve, or can microbes originate from nothing? So do we have to have an original living organism? Or again, do they come from just spontaneous generation? Another question was that there was a great interest in the process of fermentation, and scientists wanted to know what causes fermentation, because of course if we could understand that, then we might be able to reduce a lot of the crop losses or the wine losses that were happening at this time. Another one was that there was a great interest in what caused diseases. So individuals wanted to know what causes diseases, because if we can learn what causes them, then perhaps we can prevent them. So the last question was, how can we prevent infection and then also disease? So the study of these four questions, again, is really why we had so many scientists studying microbiology during this golden age of 50 years. If we talk first about this spontaneous generation of microbial life, scientists wanted to know, is this possible? So if we talk about where does new life come from, there were basically three theories that were out there. So the first one was that new life originates from other living organisms. And this is something that we know to absolutely be the case, that living things do come from other living things. But there was also a question about, can living organisms, can we get new life from past organisms, so dead organisms? If something is dead, can it give rise to something that's going to be living? And then the last theory was, can we get life from nothing? Can we get that spontaneous generation? So one of the experiments that was used to answer this question was Reedy's experiment in the late 1600s. And what Reedy did, if you just look at the picture right here, is he took essentially 3 different jars or 3 different flasks, and he placed a piece of meat in each one. One of them he left completely open, one of them he sealed up, and then one of them he covered with a gauze material. And then he let them sit for a while, and then he evaluated them to see whether or not you have anything living on these pieces of meat. What he found was that the sealed up ones did not have anything living on them. And the flask that was unsealed, we did have some maggots, some life that was suddenly there on the meat. And so what he said was that no, we cannot get spontaneous generation of microbial life, because when I seal up this container, there isn't anything that's growing on that meat when we investigate it after a period of time. So that was the first clue as to there not being any ability to spontaneously generate living organisms. A lot of other scientists, they did also do experiments to investigate this as well. So another experiment that was done, and one that got a lot of attention, was Needham's experiments. And his experiments were done right in the middle of the 1700s, and he looked at, again, whether we can get spontaneous generation of organisms. So what he did was he took a flask, he heated it up, and it's closed up, it's sealed up. He heated it up, he let it sit for a while, and then he went back, looked at it, and what he saw was that after a period of time, there was something growing inside that flask. And so his conclusion was that no, actually we can get some spontaneous generation of life. And so this stimulated a lot of interest again and this question, wondering, can we get spontaneous generation or no? Does it have to come from other living organisms? This was conclusively looked at by Spallanzani. What he did was pretty much repeat the experiment that Needham had done, but he did it more carefully. And what it was discovered is that these up here, when we're looking at Needham's experiments, these were not good seals that we were getting here. So these were like corks, and so they weren't sealed up particularly well. And the idea was that probably something was getting in there. And the reason why we think that that could have been the case is because if you look down here in Spallanzani's experiments, he did a set with no covering on them, and then he did a set with a covering on them, or with a seal. And what he found was that if he did actually seal it up, then he didn't get any growth of microbes. So nothing was there at all, which would indicate that there's no spontaneous generation. However, if he left it open to the air, then after a period of time, he did have some microbes in there. So his conclusion was that it's something in the air that's coming in that is causing the microbe growth, which means that we could have some microbes in the air that are coming in and then growing inside that flask. So these experiments basically showed that, yes, we do think that you have to have living organisms, some source of living organisms, whether it be the air, but that is going to give rise to new microorganisms. This was continuously studied until finally Louis Pasteur in the 1860s really did a nice, elegant experiment looking at this. And what he did was he used these fancy flasks that you see right here, and they have this kind of curved neck on them, or this curved tube on them. And so he took a liquid and he boiled it extensively so that he was sure that he actually killed everything in there. And when he boiled it, notice that this is going to push the steam out. The steam is going to travel down and then back up, so that the steam actually exits that particular tube. And then after he heated it up for a while, he just let it sit for many days, and what he found was that there was no growth in there. And when he did this without leaving it sealed, if he opened it up and then he waited, the microbes then grew within that flask. So what it was determined was that the air getting in is containing some microbes, and that entering air is providing a source that can then grow in the flask and produce new ice and produce new microbes, which are going to cause it to get cloudy. Up here on the top, even though it is actually open to the air, there was no growth. It's believed that the air comes in, and then any particles are really going to kind of settle down here at the low point. The particles are not going to move up the tube and then fall down into the flask. So just the length of this and the curve of this is going to keep those air particles out of the particular flask and out of that growing media, so that we don't see that cloudy mixture with the microbes in it later. So once these experiments were done, it was pretty well accepted that you did have to have microbes to give rise to other microbes. So no, spontaneous generation of microbial life is not possible. The next question was fermentation, and they wanted to know what causes fermentation. There had been many losses in the wine industry, and so they did want to have more information about this, understand how it actually worked. Louis Pasteur continued his experiments. He looked at this process of fermentation, and he set up a number of different similar experiments to try to pinpoint exactly what was causing the fermentation. So he looked at whether you can get fermentation in a sealed up flask. So that would be spontaneous generation. And just like he concluded previously, there was no spontaneous generation of fermentation inside that sealed up flask. He then looked at whether the flask can ferment if you just have air there. So if you heat up the flask, and I know it's hard to see in these pictures, but there is some heat being applied right here. If we heat it up and then we leave it open to the air, we get no fermentation. And again, that's probably because the air comes in. Any debris, particles, microbes that might happen to be there are going to settle in the bottom of this tube and won't be able to travel up the tube and down into the flask. So if he just left it open to the air, but he used that fancy curved tube, there was no fermentation. If he heated up the flask and then he left it open to the air, and we just have this straight up opening, what he found was that there was fermentation now. So this was the first of the experiments that actually gave him the fermentation he was looking for. And then he also looked at it again down here. He heated up the grape juice and then he sealed it up real nice. And if he put some yeast in there before he sealed it up, there was also fermentation. So Louis Pasteur, he did pinpoint the fermentation process to microbes. He said that microbes, specifically yeast, can result in fermentation, and that fermentation could result in the production of alcohol. So he did a lot of work with microbes. He did a lot of experiments. He solved this fermentation question that a lot of people did have. Once he did that, the next question that a lot of people were interested in is what causes disease? Because of course there was a lot of disease around them, and they wanted to know what was responsible for that. And they noticed that if people got the same disease, they had similar symptoms to each other. So the idea was that maybe they have the same microbe causing the exact same symptoms in them. People began to think that if bacteria are capable of spoiling wine, then maybe they could also be responsible for causing diseases in humans or in other animals. And so that was the thought that a lot of people were having, and this led to the germ theory of disease. And so the germ theory of disease was that there are microbes or germs, and they are causing people to have different diseases, which then have different symptoms. Robert Koch is the lead scientist that worked on this problem and studied this problem. He came up with a lot of great discoveries, and he, again, is the lead scientist in this overall field that we call etiology. Etiology is looking at disease causation. So what causes diseases? And again, he made a lot of great conclusions along this process and towards really uncovering the mechanisms and the basis of a lot of diseases. Robert Koch, first off, he developed a method for isolating specific bacteria colonies. So he created a technique for basically streaking the bacteria onto a petri plate, which is what you see right here. And then he would let that petri plate grow for a period of time. And then he would see these isolated little circles that are being pointed out right here, which represent different colonies of bacteria. And notice they're not all the same here. Different bacteria have different characteristics. You can see that some of these are very smooth on the top. Some of them are a little more blue. Some of them are a little more yellow or orange. So they have different colors. They also have very different rates of growth, as you can see by some of them having tiny spots and some of them having much larger spots. So that was one of the techniques that Robert Koch came up with, was just isolating these individual colonies of bacterial cells. Robert Koch is credited with identifying the cause of anthrax and tuberculosis. He and a lot of his colleagues made a lot of great advancements and accomplishments during this period of time. And some of these accomplishments, besides the technique where he isolated a single colony, this did include staining techniques. So techniques that they could use to stain bacterial cells so that they are visible or so that we can distinguish them from one another. There were some of the first photographs of bacteria and diseased tissues. He came up with some techniques to estimate the number of bacteria in solution. Another one was that these individuals were the first to use steam for sterilization. And then they also used petri dishes as containers for growth media and ultimately bacterial cultures. So this group, they did a lot of great things that we have built on even to date. Some of this we still use. We use some steam for sterilization. We use petri dishes for media. And we do use some of those staining techniques even still today. Another kind of lasting accomplishment of Robert Koch is that he came up with what's called Koch's Postulates. Koch's Postulates are steps that should be taken for proving the cause of infectious disease. So basically determining what causes a particular disease. And so if we go through these postulates, he said that, okay, if you want to prove what microbe, if there's a microbe, causes this particular disease, this is what you need to do. He said, first off, the suspected causative agent, so we will call something that is capable of causing disease, we call it a pathogen. Okay, this is going, I'm going to put here, it can cause disease. So the suspected pathogen must be found in every case of the disease, and it must be absent in healthy host. Okay, that was step number 1. After that, he said this agent or pathogen, it must be isolated and grown outside the host. So you need to isolate it in the laboratory. Then he said that when the agent is basically introduced into a healthy, susceptible host, the host must actually get the disease and show some symptoms of the disease. So basically what number 2, if we go back to that one, he said first you need to get the microbe or get the pathogen, the suspected pathogen, and once you get it, you infect a healthy individual or a healthy host, and then that host, we expect to get that disease. And lastly, he said the same agent must be found in the diseased experimental host. So once we have caused the disease in the host, we should be able to find that agent back in there. Koch's postulates, these are things that we still use when we are trying to prove what caused different diseases. Another big accomplishment of this day and age, the microbiology golden age, was gram staining. Gram staining is a technique that we use stain, and that stain helps us to distinguish different groups of bacteria based on their cell wall structure. So based on their cell wall structure, some of them will stain purple or dark blue like you see here, and some of them will stain red. And so we can divide them into different categories. We call them gram positive and gram negative. And those different categories will then tell us a little bit more about the structure of a particular bacteria. So gram staining is a very important technique that's still used in microbiology labs today. So all of this was important for study because individuals, even today, were interested in this question, which is how can we prevent infection and disease? So we want to know what causes it, but then we want to know what can we do about it? How can we get rid of it, prevent it? Well, one of the big things that was studied was that hand washing is going to prevent infection and disease. So there were hospitals, there were doctors and nurses that noticed that if you wash your hands a lot, then there is going to be less incidence of infection and disease. So this is something that has been very much incorporated into the way that we approach a variety of careers and everyday situations, even today. Washing your hands helps to eliminate different pathogens or microbes. Antiseptics are important as well. So antiseptics, they began to be used when we were performing surgeries, needed to clean things up a little bit. In addition to that, just hygiene in general. Keeping things clean all of the time in itself is going to prevent a lot of infection and disease. So hospitals, healthcare facilities, they were seeing a lot more hand washing, antiseptics, and then also hygiene. If we shift gears for just the last little bit of this, microbes today, if we talk about where we're at today, we have come a long way. So much has been accomplished since that golden age of microbiology. We have great techniques, great tools that we can use to study microbes, and a lot has been learned about those microbes. So today, microbes, we're still studying them, but what we know is that microbes can be used as model organisms for studying a variety of things. They can be used for studying metabolism and biochemistry, so different chemical reactions. And these aren't just chemical reactions that would occur in bacterial cells or tiny little microbes, but we can extract this information and use it to tell us something about the metabolism and biochemistry that happens in, say, the human body. So we can get information on the human body by using the microbes for study. Another one is genetics. We can learn a lot about genetics using microbes. Microbes have a very fast generation time. It's easy to grow a whole bunch of them, and they don't take up very much space. So that makes them really nice test subjects to use when we are trying to conduct experiments. Besides genetics, molecular biology, which is studying what genes do, how they function, that has been something that has been greatly studied in bacteria, and that information has also been extracted to bigger organisms, more complex organisms such as humans. And lastly, besides being able to use microbes to study a number of different things, they are also used in recombinant DNA technology. Recombinant DNA technology is technology that allows for genetic engineering, manipulation of genes in a variety of organisms. They are also used for gene therapy. So they become vectors that we can use to deliver gene therapy. In addition to that, we have microbes, of course, all around us. Microbes are used also for bioremediation sometimes to help clean up areas that do have a lot of oil spills or chemical waste that happen to be there. So they're found all around us. They can cause devastating diseases, but at the same time, they also have great benefits that they can provide to humanity.